Yeah, so much more to talk about, but I have a feeling that we should probably move on to Dr. Sumner. And if I could just give a brief introduction. So Dr. Sumner got his bachelor's degree at Cameron University in Lawton, Oklahoma, and then his PhD also from Institution, Oklahoma State University. He then took a detour to Texas, Texas A&M University, where he was a manager of the Applied Mass Spec facility and also associate director of the lab for biological mass spec from 1993 to 1999. And then he moved back to Oklahoma to join the Samuel Roberts Nobel Foundation, where he rose through the ranks as a professor in analytical biochemistry. And then in 2016, he was finally lured away from Oklahoma to become the director of the Missouri Metabolomics Center and also professor in the Department of Biochemistry at Mizzou, the University of Missouri. So Dr. Sumner has been a leader in metabolomics development across a wide range of applications, although I guess he's going to talk specifically about plant applications, which are the focus of much of his work. He's also been the past president of the Metabolomics Society and also past president of the Phytochemistry Society of North America. And he's a lifetime honorary member of the Metabolomics Society. So really a very prestigious speaker that we have in our symposium today for you, talking on integrated metabolomics for gene discovery and natural product pathway elucidation.
All right. Thank you, Sally, for the introduction. It's very flattering, and I appreciate that. So can you hear me and see my slides okay?
Yeah, yes, it's all good.
All good. Okay. So first, a comment about plants and what they eat. They may have a limited diet, but they can't get up and run away. So plants have turned into master chemists, and they make all of these fabulous defensive compounds and natural products to help defend themselves chemically and help with communication in their rhizosphere and all of those things. So that's really what I want to talk to you about today. And that's specifically how we're using integrated metabolomics to better understand, to discover these pathways and to elucidate these pathways in plants. And so I specifically asked to talk about plant applications, and I'm going to talk with you specifically about triterpene saponin biosynthesis in Medicago. This has been a central theme of our lab for many years, and so it has involved many individuals that are listed down here by name as co-authors on this work today. And so with that, let's get started. I'd like to first give you an outline of what we're going to talk about. And really, I want to introduce you to some of our basic metabolomics technologies.
Sorry, Lloyd. Your slides seem to be cut off, at least on some screens in my lab. So, for example, yeah, I don't know why.
I think I see the same thing on mine. Let me try to reshare. Okay, you probably see, okay, let's now, maybe if I go large, and then I do that, how does that look now? Do you see my full screen, or is it still being cut off?
Now is better, now is good.
Okay, so
now is good.
Yeah. I don't know what happened, but the little green box that outlines what's showing was, as you were saying, looks like things might have been cut off. So again, quickly, this is an outline of what we're going to chat about today. I'm going to tell you about our basic technologies. I imagine much of this is going to be very remedial, but I think it's appropriate for a workshop and for those trying to learn, and just to kind of re-emphasize the tools that are being used. Then I'd like to talk with you about triterpene saponin biosynthesis in the model legume Medicago truncatula. This is a very close relative of alfalfa, which is an important forage legume. Then specifically, I'd like to give you several examples of how we're using integrated metabolomics for pathway discovery and elucidation. And our basic to our premise in our experimental flow is that we're exploiting genetic and related metabolic diversity for discovery. And this is being done by metabolomics correlated with gene expression analysis, as well as metabolomics in association with genome-wide association studies. And these give us potential leads on candidate genes that might be involved, and we use traditional molecular biology and biochemistry to actually functionally characterize those. But the issue of identification has come up, and thank you, Oliver, for the introduction here. And this is something that we run into quite often. And when we see differential changes, we have to identify those molecules because that's where the biological context starts. And so we've done a lot of work in this area. I'm going to talk a little bit about our work in developing and integrating LC with mass spec and automated solid phase extraction for automated purification and concentration, and then taking that on to small amounts onto NMR for higher throughput and more confident type of identifications. So again, start with the basic technologies. And I always tell people that metabolomics is not a singular experiment. And by definition, metabolomics is a large scale profiling of metabolites. And so there's not a singular tool to do all of that. And so it's mainly due to the chemical diversity and diversity in physical properties of the different metabolites. And we, like many others, use a divide and conquer type of strategy where we use different solvents to partition metabolites into classes that have more similar properties. And so in an aqueous extract, we get the ionic components as well as metals and salts. And historically, we've done ion chromatography on these to do a non-targeted type of analyses. We are still in conversation and still trying to get ICP-MS involved in our program to do those as well. In that same aqueous extract, there are also another large number of polar organic molecules, and these are amino acids and organic acids and things of that nature. In the methanol, we typically pull out things that are intermediately polar, and this is really where many of our natural products come through, these being flavonoids, isoflavonoids, terpenoids, glycosides, things of that nature. And then at the chloroform fraction, we get our waxes and lipids. And so we then take these fractions and then go off to parallel profiling technologies, typically using GC-MS for the GC polar and GC non-polar, and typically use UHPLC-MS for our natural product analysis. We do have other non-targeted type of approaches. For example, we do a non-targeted SPE-NMR, solid phase microextraction, coupled to GC-MS for volatile analysis. We also do non-targeted analyses. And for example, we use LC triple quads to do broad profiles of hormones. And so all of these come together to form our metabolomics platform. And there was several comments about trying to keep things simple. And my colleague Gary Patty over at WashU in St. Louis always puts forth the idea, if you can do a more simple experiment, then you should be doing it. But typically in metabolomics experiments, this is more of a discovery platform, and we're trying to see as much as we can. So this is our standard workflow. We've used it for many years. We always start typically with dry lyophilized material that precipitates enzymes, reduces enzyme activity while we're handling material. We still typically start with an 80% methanol extract. Half of that we partition out and go over to our LC-MS for natural product profiling. To the remaining fraction, we add water and chloroform. This ultimately forms two phases. We get phase separation here of the water methanol, which contains the polar components. And then we have a chloroform fraction that contains the lipophilic. And then we do GC-MS on these after derivatization. And this might be GC quad or GC QTOF type of approaches. And so when we do these experiments, we get multiple data sets. And again, I apologize if this is remedial, but this is just a reinforcement of the concepts that, that we're doing more than one experiment, trying to get a broader coverage. And so in these, we get a polar extract, and this is kind of what it looks like. We get a lipophilic extract. There's lots of peaks in there. And I hope that you've talked about deconvolution and worked through some deconvolution tools. because that takes the number of peaks that we can see in the total ion chromatograms and separates them out based on selected ion signatures that are associated with them. And when we do that, we get greater specificity in the relative quantification of these molecules, but we can also resolve things better based on their unique mass signatures as well. So each of these typically, 500 to 600 components that we see in these profiles. And I believe that GC-MS gets you the biggest bang for the buck out there. And so over the years, we and the community have built libraries, and we've been using these libraries to identify compounds in these profiles. This is not a comprehensive list, but these are often types of molecules that we see in plant samples in the polar cell, polar fraction, and we see a large number of organic acids, amino acids, sugars, monosaccharides, disaccharides, sugar alcohols, amines, polyamines, and nucleotides. And so there's not a test, but this really does give you a glimpse of what can be seen. And again, I imagine this is very remedial, and you've talked about this, but that is a take-home message. With GC-MS, we can get quite a bit of coverage of primary metabolism. But over the years, we've spent a lot of time looking for and at plant natural products, and these compounds typically degrade in the GC-MS. So we've had to turn to LC-MS for these types of analyses, and we've invested heavily over the years in developing advanced high performance methods using ultra high pressure liquid chromatography. And with this comes increased resolution, peak number or plate numbers, which is also referred to as efficiency, and as well as increased peak capacity. And these are just chromatography metrics, which we use to quantify the performance of separation systems. And our current method typically has over 500,000 plates per meter with a peak capacity about 400 or 500. And that allows us to look in greater depth at the metabolome, if you will, using this one technology. We also couple this with QTOFs, and this gives us increased mass accuracy, and combined together, again, it gives us opportunity to look at the metabolome and natural products with a greater depth of coverage and greater resolution. And I often like to, illustrate this resolution and the value of resolution through a series of images. And so this is an image at low resolution. And if we kind of look at it after a couple of mojitos, then you might be able to say this is a dog. There's one eye here, one eye there, and both nostrils there. But we readily realize as we move to higher resolution that we probably shouldn't have had so many mojitos, and it's not actually a dog, it's actually something else. And it looks like maybe a group of people standing around the table. And as we continue to increase that resolution to higher and higher, we begin to differentiate individuals. We can tell often, dark hair from light hair, short hair from long hair, from no hair, and then males, females. And again, that's the concept with higher resolution in both the chromatography and mass domain. It gives us greater ability to differentiate more and more of the metabolites. And we still need a lot more depth of coverage because our approaches still are far from comprehensive, but we've advanced this quite a bit. And again, using ultra high pressure liquid chromatography and mass spec. So we've also built databases and libraries of natural products, and these are types of compounds that are in those. We've released, I believe, almost all of ours, if not, well, I think all of it to the various databases, including MONA and HMDB. But these are the types of compounds that we were able to get standards for and add to our database. And these includes a large number of phenolic compounds, a large number of benzoic acid derivatives, large number of flavonoids, isoflavonoids. There are sugar conjugates of those. We have a large number of triterpene saponins, and we actually did add some glucosinolate data to our library as well a long time. So take home message is we use these libraries for matching to identified compounds that are in our profiles, but often we are not able to identify a large portion of what we do see. And I'll talk a little bit more about how we handle that in a minute. All right. So that's kind of an overview of our technology approaches. Again, very remedial, but we kind of wanted to reinforce that. Would like to move on now to introduce you to a class of compounds called Saponins, or more common, often called triterpene saponins, and these are very abundant in legumes. And they're very structurally diverse and amphipathic. And this amphipathic or detergent nature comes from this hydrophobic C30 backbone that is then functionalized around that ring system or skeleton, and then glycosylated around at the various functional groups to give these polar side chains. So you have a hydrophobic backbone, a hydrophilic side chains, and therefore you get things that are very detergent-like. And so that's why they're called Saponins. They're kind of soapy and will foam when you shake up solutions of those. These are interesting compounds, they've been associated with a large number of biological activities. And this is a primary driver for trying to understand how they're synthesized in plants, because we may want to engineer those properties and make them better. They've been associated as anti-feedants in chewing insects. They've been shown to be antimicrobial and will combat bacteria and fungi. These are often secreted in the soil as antimicrobials, and they're also considered alleliopathic, meaning that they will actually alter growth patterns of neighboring plants in the rhizosphere. They've also been shown to influence nodulation, and this is especially important in legumes, which do fix nitrogen through these specialized organs called nodules. Many plant natural products have also been linked to many nutraceutical and pharmacological activities, and these are no different. They've been shown to be anti-inflammatory, anti-cancerous, and they induce apoptosis in certain cancer lines. They've been shown to lower cholesterol as well. And so plants, again, are great chemists, and they make a lot of these things, and we as humans have learned to exploit them over the years. But I come from an agricultural background, and this agricultural viewpoint tells us that triterpene saponins are major anti-feedants, and because of their antimicrobial activities, they cause severe disruption of the gut microbiome, and this ultimately can result in substantial weight loss or lower weight gain and performance on animals. And so we want to try to understand how these are made in planta so that we can reduce these anti-feedant properties and potentially take advantage and retain the antimicrobial or other beneficial properties that might be there. So we want to know how it's made. And so we do know that terpenes are quite ubiquitous throughout the plant kingdom, and there's various types of terpenes. These typically start with mevalonic acid or methyl erythritol and lead to the synthesis of isoprene pyrophosphate. This is a C5 building block of all terpenoids, and this then undergoes a series of condensations to make C10s, which are the monoterpenoids, C15s, which are the sesquiterpenoids. Not illustrated here are the C20 or diterpenoids, and then we're specifically going to talk about the triterpenoids here as well. So again, terpenoids are ubiquitous, but we know that isoprene yields farnesyl pyrophosphate. This undergoes a head-to-tail condensation to give us squalene. This is a C30 molecule now at this point. It's then oxidized and then cyclized. And so cycloartenol synthase is the entry point into phytosterol biosynthesis. And this illustration here is cycloartenol, excuse me. But over time, other cyclases has also evolved, and specifically beta amyrin synthase is one here that's been characterized in many plant systems. And this is the entry point into the triterpene saponin biosynthetic pathway, which is illustrated in greater detail here. So from our early profiling, we could actually see that there were many derivatives of beta amarin that correlated to functionalization around the central skeleton. And these typically are done by P450s. And so we could hypothesize that these were the responsible for various reactions that might be there. And then we then knew from our profiling that there's a large number of glycosides, derivatives, and conjugates of these molecules. And so we've hypothesized that there had to be many glycosyl transferases or multiple glycosyl transferases associated with the biosynthesis of these. So how do we go about then discovery and defining which specific P450s and GTs are involved in these biosynthetic pathways? And so the way that we did that is we began to exploit genetic and metabolic diversity. And you know, that like humans, there's lots of different individuals, many different species. There are probably 1,000 or 2,000 different individuals in the germplasm collection for Metacogo truncatula. But trying to profile all of those when we first started was somewhat of a challenge. And so we began to think about core collections. And essentially core collections utilize some type of genetic marker or genetic analyses to try to define the genetic diversity. And so over time, several groups have made these core collections, including my former institution at the Nobel Foundation, who, by the way, have closed their scientific doors as of June, which was kind of sad to hear. But also the French group created a core collection of 192 lines. And the idea is through the genetic marker analysis, they could quantify the genetic diversity and say that these 192 lines represent 99% of the genetic diversity, and hopefully the associated metabolic diversity as well. So we took these core collections and we began to profile them, and specifically looking at natural products. We also did primary metabolic profiling of these, and that data is available. But we began to ask the questions then, can we identify lines that have high levels and low levels of the triterpene saponins? And if so, we could then go in and do association or correlated gene expression analyses using microwaves or RNA-seq type of experiments. As time has evolved, many of these lines have also been sequenced as part of a HapMap project. And when that was done, that enabled us to do genome-wide association as well, where we associate total saponin content or individual saponin content, i.e. the metabolic phenotype, with polymorphism in the genomes. And that, again, helps us identify putative genes involved in the biosynthesis. And so we have both correlated gene expression analysis and this GWAS information to help us first discover and then potentially prioritize the candidates, which we then take on to doing traditional molecular biology and biochemistry, which is a little bit slower but necessary for proving the function of these specific genes in specific metabolic pathways. All right, so we grew these plants out, and this is just an image of the noble core lines. And if you start looking real quickly, scanning across the various lines, you can begin to see differences. And there's differences in leaf morphology. Some have serrated leaves like these two, but some are smoother. Some have trichome hairs all over the leaves, others do not. Some of them have these purple anthocyanin spots, others do not. And this is actually a visible chemical phenotype because anthocyanins are a part of the phenylpropanoid pathway. But unfortunately, we couldn't see the triterpene saponin content. So we actually had to do the chemical analysis, i.e. the LC-MS type of profiling of those. And this is just kind of a summary slide of some of the lines that were involved. These were collected from various geographical locations around the Mediterranean. We then went in and profiled both the aerial tissues and the root tissues. And in these profiles, we could see various peaks. We then did tandem on these to try to do identification of many of these, and we'll kind of come back to that. You know, we can identify maybe over 100 of the different triterpene saponins at this point in time. So the question is, was there, you know, heterogeneity in the accumulation of saponins, where they're hyper and hypo lines. And we have to look to the metabolic profiles to do that. And these top two are chromatograms for different leaf profiles, and these bottom two are different roots. And we can quickly see that in this red box region, this is the dilution region of the triterpene saponins, some lines had very low levels relative to others that have very high levels. And these are normalized to the same Y scale value here to giving you a view of the relative expression of those or accumulation of these metabolites. And so we see difference accumulation in aerial tissue, but we also see differential accumulation in root tissues, where this line has just a tremendous amount of triterpene saponins relative to the others. We could do cluster analysis on all of those, and we could begin to associate that with geographical origin, but we really want to focus on the biochemical synthesis of these. I want to make one other comment, though, before we do that, and that is that there does seem to be tissue specificity in the accumulation of the triterpene saponins. So, for example, xanthic acid okay, sorry, I heard an interruption. Xanthic acid seems to accumulate highest in aerial tissue and very little in root tissues, whereas some, such as biogenin, accumulate predominantly in the root and very little in the aerial tissues. And so these are suggestive of ecological roles. For example, xanthic acid, we believe, is the primary anti-feedant, if you will, that's associated with aerial tissue and herbivory, whereas biogenin and these other root-specific soy sapogenol seem to be more associated with antimicrobial activities in modifying the soil microbiome. Okay. So we did the profiling, and then we began to say, okay, can we identify lines that have differential accumulation that would be good for gene expression analysis? And in this process, we just summarized total saponin content. You could summarize and look at total aglycone classes or even at individual molecules. But we first started with total, and we selected a couple of lines. And for example, we selected this GRC line because it had the lowest level in root tissues, but relative high levels in aerial tissues. And vice versa, this ESP105 line had the lowest in aerial tissues and the highest in root tissues. And we then picked a couple of reference lines, for example, A17, which was the genotype that was first sequenced, and we used it as a reference. And so when doing these experiments, you could then do a correlated gene expression analysis. We selected various lines that would allow us to look for correlations and contrast across lines. For example, A17 has a high level in the aerial tissue, but this ESP105 has a very low level, so we could contrast it there. But we also selected lines that had differential tissue accumulation of these, and so that we could do correlated gene expression based on tissue type as well. And ultimately, this gave us a multi-factorial type of correlation network, which we could prioritize genes and gene candidates to take on to the next step. And so there's various ways of doing the correlations. One is you can do a simple gene-to-gene correlation with the expression analysis. And this is just a correlation heat map of that. Above this diagonal line is the correlation coefficient. And as it moves towards 1 or unity, this becomes darker red. And we see kind of two areas of very high correlation here in this little block area and here and down here. And below the diagonal is a correlation or a p-value saying that these correlations are very strong, but also very significant. And if we look at this one little cluster here, we can see readily that many of our early enzymes involved in triterpene or in terpenoid biosynthesis are present, things associated with mevalonic acid, IPP, beta-amarin synthase, but we also see in this cluster several P450s. And lo and behold, the long story short is that these have now all been proven to be functionally associated with the biosynthesis of triterpene saponins. And so we can do gene-to-gene correlations. We can do gene-to-gene metabolite or metabolite-to-gene correlations as well. But again, this is just a discovery platform for that initial association. And then dependent upon how many times or the strength of that correlation, we began to prioritize candidates for characterization. And so typically we use biochemistry to help improving this function. We would clone these genes. We would express them in a yeast WAT11 cell culture system. We grow these out, we'd isolate microsomes, and then we would feed those potential substrates and ask the question, do they make any other triterpene saponins? And the answer to that is yes, we had several successes in this area. And so this is an example of a very highly correlated CYP72A68. When we have different profiles here, we have the empty vector plus cofactor on top, and then we have the overexpression of the CYP72A68 plus cofactor on the bottom. We look at the empty vector, we fed it oleanolic acid. We really don't see much in the way of products there. But with the overexpression of CYP72A68, we see multiple products that come up in that in vitro biochemical assay. And when we identify them, we could begin to see the functionality of this enzyme. And we could tell that it was a C23 multifunctional oxidase, and that we see multiple oxidized versions of this. We see the hydroxylated version, we see the aldehyde version, and then we see the carboxy acid of that. And so this, plus the molecular evidence that we had, told us that this CYP72A68 was, again, a multifunctional oxidase associated in triterpene saponin biosynthesis. Specifically, it was a C23 multifunctional oxidase. So we had other gene candidates as well. We had a CYP72A67. And again, these are the in vitro bioassays. The top is the empty vector plus cofactor. The second traces are the overexpression minus cofactors, and the bottom traces are the overexpression plus cofactor. And we used multiple substrates in this. Well, actually, we tested many different substrates, and including flavonoids and isoflavonoids, things of that nature. But these are the two that showed some activity here. And so when we fed this CYP72A67 oleanolic acid, we could see that a new compound was made, and this is had been identified as a 2-hydroxy oleanolic acid. And then we also fed it heterogenin, and we got another 2-hydroxy version of heterogen called beagenin. So we didn't just rely on the biochemical, in vitro biochemical assays. We also looked to advantage and proved this using molecular approaches as well, including RNAi of this CYP72A67 in Medicago truncatula hairy roots. Here is just the expression levels of the various control lines, and these are the RNAi or RNAi interference lines showing reduction in the gene expression of this specific 72A67. And when we look at the chemical profiles, we could actually see, when we knock that out, we could see increases in what we believe are the substrates, and this is oleanolic acid. And if you remember from previous studies, this 72A67 makes the 2-hydroxy version of that. And when we knock it out, this the substrate increases, the product decreases. And same thing with heterogenin. When we knock it out, we see an accumulation of the substrate and a reduction of the product, and therefore supporting this as a 2-hydroxy hydroxylase of triterpene saponins. All right. So those are examples that came out of the correlation analyses. We also had GWAS data, and we began to ask the question, is there any correlation with specific loci or polymorphism in various loci on the genome that would also correlate with triterpene saponin content? And this is what's called a Manhattan plot. And more or less, it's a function of the chromosome location and some probability associated with it. And this red line is the threshold line, and things about that are more significant. And when we look at that, there are many good quality leads here that we began to think about that could be involved in triterpene saponin biosynthesis. So we took one of those leads that was identified as a glycosyl transferase, and we kind of followed up on it. And I talked about this multifactorial type of prioritization. And so there is a gene expression atlas for Medicago. And so we could look again at this gene-to-gene correlation, and we asked the question, does this potential GT correlate with other triterpene saponin enzymes? And the answer to that is yes. On this left graph, you can see the correlation of those graphically. If you calculate that correlation coefficient, I think it's above like 0.95. So they're very highly correlated. And then on the right, we have this gene correlation network, which again begins to associate our unknown UGT with other known triterpene saponin enzymes and potentially other unknowns that are associated with those as well. So it stood the test, multiple tests, and held its priority, saying we needed to go characterize it further. So we took this candidate GT and we overexpressed it in hairy roots. And here on the top panel is just a view of that UGT overexpression in hairy roots. And the top one shows two giant peaks that just pop up compared to the empty vector control. And this is the total ion chromatograms, and then there's a selected ion chromatograms and box plots of all those. It's all the same data. We're just looking at it in different forms, but the take-home message is this is a night and day type of thing. And so these things pop up, and so they're associated with this specific UGT. But unfortunately, we were not able to match it to anything in our library. And this is a common problem that we face. And so, we could partially hypothesized that it was a soy sapogenol B, but we didn't understand the glycosylation pattern. And again, it didn't match with anything in our library up to this point. So how do we go about identifying these molecules? Many labs stall at this point, but we've really done a lot of work in this area trying to improve our ability to do this in a higher throughput manner. And so Arapana talked earlier about the way that we use and generate libraries and then use spectral matching for metabolite identification. But she also mentioned the problems with that, and that is you ultimately run out of money and you run out of availability. Many of the things that we're interested in are just not available commercially. And so how do we go about identifying those? And so one of the ways that we've gone about that identification process is trying to understand the data that we have. And we've built computational prediction tools that will help us identify potential molecules from their LC-MS and or EI-CI data points. But ultimately, these are predictive tools, and we really need to prove the structure if we're really out to understand the biochemistry. And so we've built other tools, empirical tools or experimental tools that allow us to do automated purification of our target analytes, coupling LC with mass spec and online solid phase extraction. And once we get enough material through repetitive injections, we then take that over to our NMR, and I'll describe these in more details. Okay. So first thing is, again, we try to predict the identity of the molecules that we have with computational tools and data that we already have. This is the work of Dr. Fong Chu, who's now at International Flavors and Fragrances in New Jersey, but he built a tool called PlantMat, or Plant Metabolite Annotation Toolbox. And it works on the LC-MS, and this really is focused on natural product types of identification. And he also built a module that works for GC-EI data as well. But I'm really going to focus on the LC-MS side. So there are tools out there that you can, and there's many a database out there that you can take your tandem data to and search it. But for that to be successful, that molecule has to be in that database. If not, you're just getting back false positives, and we actually have spent a lot of time chasing these false positives. So we began to think, could we be smarter about this? And could we use what we know about phytochemistry to maybe help with our success? And so what we ultimately decided to do is we knew that most natural products have some type of centralized skeleton. These are then functionalized at various points or around that skeleton. They are often glycosylated and they're acylated. And so we treated these units as different building blocks, i.e. little Lego blocks. And then we put them together in all combinatorial possibilities, and then we searched that space. So these are not known metabolites that are there, but they're possible ones just based on informed phytochemistry of what might be the. And when we started doing that, we started having great success. And so this is an example of that process in that we do the profiling. We see a molecule, then we measure its accurate mass, and then we can take all those different Lego blocks together and then just start looking at them in a combinatorial possibility to see which one might match to this accurate mass. And when we do that, we start getting multiple typically hits that are associated with that potential identification. Now, although we've not uniquely identified this molecule at this point, we have reduced the chemical space quite dramatically from a truly unknown to a series, for example, of different triterpene saponins that might be involved. But we also have the tandem data for that. And, so this is further illustration that we have an accurate mass. We run the plant met, we get a series of identifications or putative identifications for that. But we can also then go on and do the tandem experiment and then search this tandem data, the real experimental data for our unknown, against the potential fragments of these other potential identifications. And there's other tools out there, other tools out there that you might want to use for that. So specifically METFrag, for example. And so we really just start correlating the number of identifications or the number of fragment ions in the spectral data to that which we would predict that would be there. And the idea is that more components or ions that are correlate, the higher the confidence that that is likely the identification. And we ultimately can reduce the complexity of our list, but ultimately we're left with an issue of stereochemistry. And if you look, all of these have the same mass error. And when we look at these potential IDs, they're again just stereoisomers of those. And so again, we're stuck with the question of what is the stereochemistry of that aglycone? What are the stereochemistries of the sugars? And where specifically on that molecule are those sugars? And this is often an area where mass spec fails, and that is in the area of isomer analysis. Sometimes we get lucky and we can differentiate these by retention time. Sometimes we get lucky in that the fragmentation is unique enough, but many times we are still unable to resolve these confidently. And so that's why we've invested in this complicated or integrated system that couples LC with mass spec, online photodiode detection, solid phase extraction, and then NMR. And these are the colleagues that have worked on this system over the years. The most recent is Raj and Dr. Gosher. So the way this works is we do our extract, we do our comparative analyses, our LC-MS profiling. And when we find things of interest, we have the accurate mass and things that we can predict an identity for, but then we want to purify that in a higher throughput automated manner for NMR. And so the way we do that is we have software tools that will allow us to divert that flow onto a solid phase extraction. And this is fraction collection. And I imagine many in the crowd have actually collected the unit flow from an HPLC column, but instead of collecting it into a test tube, we're actually collecting it and reabsorbing it onto a cartridge. Now, mass spec is much more sensitive than NMR, so we have to get enough material to get a good signal by NMR. And the way that we do that is we repetitively inject the sample and collect that same peak on the same cartridge over and over again. So we'll trap from 20 or 30 injections until we get somewhere about 1 to 10 micrograms, and that's usually enough for a good proton and maybe for a good 2D type of spectra. So once we have it trapped here, we dry it with nitrogen. We then elute that with a deuterated solvent, and then we take that over to a special NMR. And this is a 600 megahertz instrument with a cryo microprobe. And again, If we get 1 to 10 micrograms, usually we can get a pretty good proton spectra, 1D proton. If we get towards that upper end of that, we can actually get 2D spectra associated with those. These are typically HMBC, HSQC, or other, indirect carbon type of detection methods. So that was how it a cartoon of how it works. This is actually the instrumentation in the lab. We have our UHPLC here. Here's our column. We have an online photodiode array detector. And then we have this eluent come over to the ionization source of the mass spec. But prior to that, there's a splitter that only allows about 3% of that eluent into the mass spec. The other 97% come over here to this spark column or Bruker Prospect 2 SPE device, and where we trap these onto these SPE cartridges. And this actually has 2 trays of 96 cartridges, so we can actually do this in a highly parallel manner. We never work at capacity, but we often will set up separations and trappings for 20 or 30 compounds over the weekend. And so once we have them trapped, we can then elute those from these cartridges. We can elute those into vials, or we can elute them into NMR tubes. For our cryo microprobe, we typically elute into vials first, and then we'll concentrate those down and then introduce them into our NMR tubes. And the reason for that is the active volume in the NMR tubes is about 30 to 50 microliters, but the optimal elution volume of these SPE cartridges is on the order of about 300 microliters. So we can't fit all of that in the tube, so we'll elute into a vial, concentrate it down, then put it in our NMR tube, and then go over to our NMR to collect data. And we are collecting data in an offline manner. Early on, I thought about this a lot. I mean, it would be kind of cool to do it online, but truthfully, with an offline type of approach, we can do a lot more experiments and do longer experiments that we could not do online. And this is a little bit more critical, increasing your signal to noise when you have very limiting sample quantities. So again, we collect our data, we identify peaks of interest, and then we set start and stop times for the trapping. So in this 5-minute window, we have 1, 2, 3, 4, 5, 6, 7 trapping events. And then once we have set those, we begin to inject over and over again. And each time we're collecting that peak onto a SPE cartridge. So we're purifying 7 components in parallel from, in this case, 20 repetitive injections. Okay, so we do that. We then take it over to our NMR, and I just kind of want to bring all of the workflow together now. So typically we do the LC-MS and LC-MS-MS types of approach, use our computational tools to begin to predict structures. And often this can give us an idea of the different puzzle pieces that are involved. It can give us some of the ideas of the aglycone as well as the sugars that are connected to it, as we saw earlier. But once we have that information, we can then begin to do the NMR to get greater confidence in the stereochemistries of these. So we can do HSQCs, which often are very helpful in identifying the aglycone, the specific structure of that glycoside. And we'll often see shifts in the HSQC depending upon where the sugars are as well. But we can do other NMR experiments. This is a TOXI, or total correlation spectroscopy. And this often is very useful in identifying the specific sugars. And it can tell us if it's glucose or galactose and the nature of those different sugars that are present. Then we can actually do long-term coupling types of experiments that help begin to put this together in a 3-dimensional organization. We also see that from the other spectral data as well. And ultimately, we have a 3-dimensional structure that we have some fairly good confidence in that structure at the end of the day. Okay, so I'm starting to wind down on time, but still have just a couple more slides. So back to our problem. We had this putative UDP glucosyl transferase that we overexpressed, and we saw these giant peaks here, but we couldn't identify it. So we did this, we took our LC-MS-SPE system, and we purified this, and then we ultimately did 1D and 2D NMR. And what you see here is a structure that we then associated with that. We were right that the backbone is soy sapogenol B. We could tell that there was a galacturonic acid at this end of the molecule, but there was also, we discovered there was a unique sugar down at this end called DDMP. And this is dihydroxy methylpyranum. It's kind of similar to maltose, if you will. And so through that identification, we could then begin to see that this putative UGT was not a UGT, i.e. it didn't transfer glucose. It transferased this unique sugar, DDMP, and we could actually tell that this put this on this soy sapogenol B, and this is the actual activity of that. And so from this, this actually has identified the first DDMP transferase node in any plant system, and just another example of how metabolomics is helping us understand the pathway. So just kind of want to put a quick plug here that we're also building tools that a couple LC-MS-SPE with a new technology called micro-ED. This is microelectron diffraction, which is a cryo-EM method. And most of the workflow is the same, except when we get to here, we have a small amount of material. We then crystallize that and are taking it over to a micro-ED instrument. Missouri has ordered one. It's supposed to be here this fall, but in the interim, we've been working with Professor Brent Naniga at the University of Arizona and trying to further enhance our higher throughput metabolite identification platform. And our hopes are that by the micro-ED, we remove a significant component of the interpretation of NMR spectra and get a more definitive three-dimensional structure directly from the crystallography. All right, so inclusion, metabolic profiling of Metacargo truncatula identified chemical and genetic diversity. We used that and exploited that to do correlated gene expression analysis that identified multiple genes involved in saponin biosynthesis. I gave you a couple examples of P450s that are associated with that, but also gave you a really nice example of a unique sugar transferase that we've been able to identify. And take-home message is it works. And this is a very focused view of a single class of compounds in legumes. But many of our peers have used similar types of approach to identify a large number of natural product biosynthetic enzymes over time as well. With that, I'm going to close and thank the University of Missouri for their funding of our lab for the last 5.5 years. Also, a lot of our work merged while we were at the Noble Foundation. And as I said earlier, I'm sad to see that they have closed their scientific divisions there now, and it's gone mainly to an applied agricultural research institute. We thank NSF MRI for our NMR. We've had a longstanding relationship and collaboration with Bruker Daltonics, ICO, Barsh and his group, Sven Myers, Heino Heinen, who's no longer there. And then we'd also like to thank NSF iOS for funding a unique collaborative opportunity with the Japanese Science and Technology offices and our colleagues, Kazuki Saito and team in Japan that helped us really evolve the LC-MS SPE-NMR system. We do have a core lab here, and if you're interested in working with us on plant-based metabolomics, we'd love to hear from you. You can contact me or my colleague, Dr. Jintian Lei. He and I have now worked together for over 20 years. And so with that, I'm going to stop and take questions.
Great. Thank you, Dr. Sumner. We do have a few questions in the chat. Do you want to go through them? Would you rather I read them out?
Let me, well, I have to go back up. Okay.
I think the first one is from
wait,
Shweta.
my-
Is your library available to other researchers?
So yes, as I mentioned, we've published this and there's been several publications. we've given the data at Oliver, and I know it's in Mona. We've given it David Wishart. I know that it's in HMDB, so much of that's out there as well. We've also been recently working on CCS libraries as well. That was recently published and is available as well. John McLean and his group has done a compendium. So we have historically been very eager to share information with others. So.
Great, thank you.
Okay, variation between different organs, the same plants, endophytes, exophyte, and not the plant. You know, that's a good question. It's very difficult to differentiate what is a plant metabolite and what is a endophyte. We know some about the genetics of endophytes, and they make certain alkaloids, and plants don't have those. But at the same instance, same breath, you know, it was often thought that trehalose come from fungal interactions, but it's now been shown that plants can produce it as well. So it's a really fine line of determining the origin and the extent of that. And you know, as Oliver said about humans, you know, one could say the same about plants, and that there are ecosystems, and there is a microbiome on the surface of the roots, and there's our endophytes, and so these come together as an ecosystem to do metabolism. Okay. Is micro-ED more sensitive in NMR? We're hoping that it's about the same. It's early on, I think it's been oversold, micro-ED, but we're beginning to see some success at a similar level, at the microgram level, if you will, nanomolar level with micro-ED. So I wouldn't go as far to say it's more sensitive, but I would say it is probably approximately the same level of sensitivity to a cryomicroprobe. But hopefully, as I mentioned, the interpretation will be easier with micro-ED. So once we get it all figured out in the pipeline for, you know, the diffraction and everything, then we just push a button and we get a structure. That's our hope. It's not proving to be that simple at this point, but that's why it's called research, and then we just need more funding there. So, okay. Oh, let me scroll down. Well, thank you for all the kind comments. So there is a question about if I have both metabolomics and transcriptomics data, do I still need to use qRT-PCR to further identify a key gene? So my answer to that is almost always, if you're trying to prove function, the quantitative precision and accuracy of, say, microarrays or RNA-seqs is nowhere near that what you can get by qRT-PCR. So that is, I view metabolomics again as a discovery platform, and that's one of the early next steps you would do, is to go to see if the qRT-CPR correlations hold as well, if not better, than the transcript data.
I could ask a follow-up question on that topic before you move on to the next question in the chat, which is that you discussed how you put to very good use expression data, in this case from microarrays, and also you did, and that can help you to identify candidate genes. And you also did a GWAS analysis where you identify SNPs that are correlated with changes in saponin content. Right. My question is, have you tried eGWAS, where you look for SNPs that are correlated with changes in transcript abundance of your key genes? So in this case, the phenotype is the abundance of the transcript of interest rather than the saponin itself.
I'm not quite sure I understand that correctly, but I thought that's the foundation of GWAS. Where you're,
yeah, so GWAS, so when you did GWAS, you looked for correlation between SNPs and saponin content, right?
Right. So,
and
or indels even, so.
Or indels, yeah. For eGWAS, you look for correlation between SNPs and transcript levels.
Okay, so we did. Okay, I think I'm so sorry. I was so no, we're not really do that, but there's.
Sounds like you could. Sounds like
yes, there are. Unfortunately, as Oliver was mentioning earlier, you run out of money and postdocs move off, and that's there's always all these additional experiments that you'd love to do, and Oliver has been much more successful in building databases and compiling all of his data in a useful manner. We've just never have had that level of support to do that. So, but the data is there, and it could potentially, we could potentially do that. So we still have another big cluster in those correlations that seem to be very hot that we have not characterized. That's additional future work in that area as well.
All right, so I think there's a question from Hannah Parks.
Yeah, so in regard to the P450 enzymes that I talked about and several substrates, the question is, have you tried this enzyme with substrates found outside of metacargo, i.e. steroidal glycoalkaloids? So the answer to that is no, but my colleague, what is his name, in Denmark or Copenhagen? Alan Goossens works in Metacargo as well. He's a much better molecular biologist than I am, and he's doing all of these wild combinatorial type of things with enzymes, trying to make new terpenoids that might have additional nutraceutical-pharmacological or benefits to the plant, and I think that's really cool stuff. So, all right.
Yeah.
Okay, I think that's all the questions.
Yeah.
Thank you again for the time and opportunity to chat about our work.
Yeah, and thank you for a very well